Okay, next we're moving on to our chemical senses. And again, just a general understanding of what's going on, nothing crazy specific. So, our chemical senses are smell and taste. When we're talking about the receptors for smell, which is also called olfaction, and taste, which is also called gustation, the receptors are chemoreceptors, meaning that the receptors respond to chemicals in some sort of aqueous solution. They complement each other and they respond to a variety of different classes of chemicals. So, smell receptors are going to be excited by airborne chemicals, and taste receptors are going to be excited by food chemicals that are going to be dissolved in our saliva. So, when we're talking about smells in general, that each smell can contain hundreds of different deodorants. Hundreds of different odorants. There are about 350 to 400 different odorant receptors in humans, or genes for these, and each receptor responds to one or more odorants, and each odorant binds to several different receptor types. However, each receptor cell has only one type of receptor protein. If we're talking about pain and temperature, receptors, those are also located in the nasal cavity, and they can respond to irritants such as the really sharp smell of ammonia, or if we're talking about the hotness of chili peppers, or even the chill of methanol. So, impulses from these receptors are going to reach our central nervous system along the afferent fibers of our trigeminal nerves. So, in order for us to smell a particular odorant, it has to be volatile, meaning that it must be in a gaseous state as it enters into our nasal cavity. Additionally, it has to be dissolved in the fluid that's coating the olfactory epithelium. So, dissolved odorants are going to stimulate olfactory sensory neurons by binding to receptor proteins in the olfactory cilium membranes. When it binds, it's going to open cation channels and generate a receptor potential. Ultimately, we're going to end up with an action potential that's conducted to the first relay station, which is located in the olfactory bulb. So, transduction of odorants uses a receptor that's linked to a G protein. The events that follow odorant binding are pretty easy to remember if you compare them to what we've already talked about. So, if you already know the general mechanisms of receptors and G proteins, which we talked about both in the nervous system as well as in phototransduction with the eye. So once we have an odorant that's coming in and it binds to a receptor, this is going to activate G proteins. When we're talking about G proteins in relation to our sense of smell, we use a subscript OLF to represent olfaction. So once we have our G protein activated, we're going to have it's going to activate enzymes. These are adenylate cyclases that synthesize cyclic AMP or CAMP as the second messenger. And then our cyclic AMP is going to act directly on the plasma membrane cation channels, causing them to open, and we're going to have the same thing happen where we're going to have sodium and calcium enter in. Because we have a positively charged ion sodium coming into the cell, this is going to cause depolarization and ultimately transmission of the impulse. The calcium coming in causes the transduction process to adapt, decreasing its response to a sustained stimulus. So this is what we refer to as olfactory adaptation, and it explains why we can't really recognize the smell of our home anymore, but if we immediately walk into somebody else's home, we can tell kind of the smell of that. Or if we've been on vacation for a really long time, when we come home, maybe we recognize how our house actually smells. So this is that pathway. Again, it's very similar to the one we saw in phototransduction. We have some sort of odorant that's binding to its receptor. Once it's bound, it's going to activate a G protein that's going to translocate across the membrane and bind to an adenylate cyclase, which is now going to become activated. When we're talking about phototransduction, it wasn't adenylate cyclase, it was phosphodiesterase. But regardless, this enzyme is going to cause activation of cyclic AMP. Once we have